I heard a lot of people are saying that the superdelegates, actually superdelegates are saying that they would just vote generally for whoever wins and not like.
You watch
that's because
that was on Stephen Colbert.
Fox News too.
Oh, really?
That's on Stephen Colbert.
That was on Stephen Colbert.
With Megyn Kelly. Oh, do you watch Fox News?
No.
Is that really no, that was a girl.
Megyn Kelly's a really attractive blonde.
That looks good.
I can't.
Is it okay if I move it around a little bit?
No,
Zoom in, you
don't,
know, and,
don't Cloverfield.
Alright, does anybody know what movie that is?
Riot People.
Does no one know what movie that is?
Looks like War of the Worlds.
It's called Soylent Green.
That's one where they eat, like people get sucked up.
Alright, it's protein! In the movie, there's this food, Soylent Green. At the end, they realize it's people! Because it's this overpopulation, they run out of food, so they make this food that people eat.
So food is people, food or food?
People is the food.
Oh, people is food.
Okay, so last week in lecture, we talked a little bit about MALDI-TOF, just that that's a technique of mass spec, and that you can use that to identify proteins. That'll come into play a little bit today. We talked a lot about protein purification, salting out dialysis, and we went through the 3 types of chromatography that you need to know: gel filtration, affinity, and chromatography, and ionic exchange. And you should know what those are, and you should know what the elution profiles would look like when you elute your protein off the column. And then we talked about SDS phase gel, how that was separation of proteins by size, and that you add SDS so that all proteins have the same type or same charge to them. And then there was something called native page, and native page was separating proteins, but separating them when they're still in their oligomeric state. When you add SDS, everything is normal. Yes.
So what is exactly the difference between agarose and SDS? Because agarose all separates the sides.
Yes. Polyacrylamide gels are a less porous material than agarose. Proteins wouldn't get very good separation in agarose. There's a better separation in polyacrylamide. Polyacrylamide has a smaller pores than agarose.
So
You can, in class, we'll actually in lab, we'll actually separate DNA using polyacrylamide, but DNA can be separated by agarose, so normally they do it that way. It's easier to make an agarose gel than polyacrylamide.
different kind of gel.
Okay, so today we're going to talk about protein-protein interaction that is associated somewhat with chromatography. It's purifying proteins and looking at proteins using the protein that they interact with. And then we'll go to determining protein concentration. How do we determine protein concentration in the lab? And then we'll go into protein structure. And so when we go into protein structure, that's going to go into class. First class after the exam is going to be in the computer lab. We're in 103 Jonas Park. And we're going to, the first class will be looking at the computer program that we can use to look at proteins like possibility of gas, things like that. And then the class after that will be also be in the computer room, and we'll look at crystal structures. We'll have a program where we can manipulate crystal structures, and that crystal structure will actually look at the T7G4 protein, and then we'll look at exactly the amino acid that we're mutating in class. Okay, protein-protein interactions. These can be, over here we have receptor signal and binding. So you have a receptor and you have a signal peptide, and they interact or to pass on the signal. Signal transduction, kinases binding to other kinases, and enzyme complexes. And the enzyme complex that people should at least be somewhat familiar now with is the repliposome, right? That's what replicates DNA. The helicase there in the middle of the replosome is the protein that we're purifying in lab, and the replosome has more than one component. It has DNA polymerase, it has single-stranded DNA binding protein, and if you were to look at these proteins, it's possible to purify these proteins together as a complex. So in that manner, you know that those proteins must interact. And they must interact at least somewhat strongly, or they wouldn't be able to compose a replosome. So the first technique we're going to talk about is tandem affinity purification, and that's going to look pretty similar to affinity chromatography, where we had nickel and we had a his tag. This way, we're using the affinity is going to be protein affinity for another protein. And then we're going to go into two that aren't really associated with chromatography at all. Something called the yeast 2 hybrid assay, and then phase display. All right, tandem affinity purification. You have a protein of interest, and that's on 2 successive tags. Okay? And so you have your target protein in this diagram, and then you have 2 tags which will bind to an affinity column. One here, shown here, is a common binding peptide, and the other one is called protein A. Okay? So you run a first column. That first column will bind protein A and leave the common binding protein alone and your target protein. And that